Hello, I am Cécile Erat. I am working for the French Alternative Energy and Atomic Energy Commission. In my lab, ProCytox, we are specialized in cytogenetic assays for biodosimetry. Today, I will explain you a tricky cytogenetic assay used to quantify chromosome break in a few hours. This assay is called premature chromosome condensation assay or PCC assay. In the lab, we perform the classic PCC assay and we improve the technique by coupling it with the centromere and telomere fish staining. I will provide you with a detailed protocol and show you the critical steps of the technique. In our lab, we perform three main techniques for genotoxic assays and we mainly apply it to biodosimetry. For each, we have created online tutorials. The goal of the different techniques is to evaluate the dose received by individuals and population after an accidental radiation exposure in order to make the triage and then treat the maximum of people. Briefly, I show you the characteristics of each one. The decentering end ring assay, or DCA, allows for the detection of decentering end ring chromosome generated by two misrepair DNA double-strand break. It requires 48 hours of blood culture. In the field, this assay is the most widespread and is considered as the gold standard. The premature chromosome condensation assay, that I will deeply describe later in this video, allows for a fast and rapid analysis without the delay of having to establish a cell culture, and enables the detection of excess of chromosome fragments. The sensitivity of this test is quite high, with a detection limit of 50 mg, while the cytokinesis block micronucleus assay allows for a fast and partially automated analysis after 72 hours of cell culture. The DCA and CBMN assays are described in two other online tutorials. This video will show you how to obtain premature chromosome condensation from G0 lymphocytes that are non-dividing and contain interphasic chromosomes. The principle of the technique is to provoke the fusion between the G0 lymphocyte and a cell in metaphases. Commonly, the hamster CHO cell line fulfills this function. This technique allows rapid sample processing and analysis of chromosome breaks without previously established a blood culture and can be used in case of crisis for fast patient sorting. On the left, you can see the result of a PCC obtained after Giesa staining. The darkest and thickest chromosomes correspond to those of hamster, while the smaller ones are the human premature chromosomes. To analyze the obtained images, the principle is to count the number of human chromosome fragments exceeding the expected 46 chromosomes. On the right, I present you how we perform the analysis in our lab. In order to facilitate and improve the analysis, we make a centromere and telomere fish staining, allowing the visualization of decentric chromosomes with two centromeres and acentric fragments without centromere. Irradiation of lymphocytes causes a linear relationship between the received dose and the double-strand break. After having built a dose-effect curve, which requires at least the data from 5 donors, we establish linear quadratic relation between the number of PCC fragments and the irradiation dose. We count the chromosome fragments in the unknown irradiated sample and we can determine the dose. With our improved technique of centromere and telomere staining, the principle of analysis remains the same, but we can directly correlate unstable chromosome aberration with dose. Here, I listed all the equipment and reagents you need to perform the assay. Please check it closely before starting. The different solutions you need to prepare for performing the assay are also listed. Note that it is important to use a freshly made Carnoy 2 fixative. A particularity of the technique is that you need to anticipate and prepare the mitotic cells the day before the assay by splitting them at about 1/3. The aim is to reach 70% of confluence 24 hours later. But don't worry, you can also freeze THO cells in mitosis and use them in case of an emergency. The next day, about 24 hours later, corresponding to the mitosis wave, add colchicine in the cell medium at 0.15 microgram per milliliter and incubate CHO cells for 4 hours more at 37 degrees. For this assay, you need to prepare both the lymphocyte and the CHO mitotic cells. Of course, you can manage preparing both in parallel. But for an easier understanding, I will present first the lymphocyte isolation using Fecal. For one fusion leading to PCC, you need about 1 milliliter of whole blood. For a small volume of blood, you can do the fecal separation of peripheral blood lymphocyte or PBL in a 15 milliliter tube. I start by adding 5 milliliter of fecal solution. Then I take here 2 milliliter of whole blood. I carefully drop it on the top of the fecal. You need to do it slowly. To make sure you don't go too fast, you can set the pipette boy at slow speed and drop the blood on the wall of the tube. Please look carefully how I do it. To isolate the red blood cells and the PBL, we centrifuge at 1800 rpm for 20 minutes without any break. You will obtain this. The red blood cell will be at the bottom of the tube and the PBL will form a white ring in the middle. With a pipette, aspire the PBL. Rinse them in 10 ml RPMI medium. You can centrifuge the lymphocytes at 1400 RPM for 7 minutes. Here, I recommend you to perform a second rinsing step, and before the second centrifugation, you can anticipate and divide the lymphocytes in round bottom tubes, calculating 1 tube per fusion. In parallel, you will need to prepare the CHO mitotic cells. The most important point is keeping colchicine into the CHO medium in order to maintain the cells in the mitosis phase. You take the CHO cells from the incubator. Then you remove 7 ml from the 10 ml of the culture medium and keep it for later use in a 15 ml tube. Using an optical microscope, check the cells. The mitotic cells present a round shape and are easily detached from the flask. To take the maximum of mitotic cells, we just heat the flask on the side of a table. To collect the floating cells, you can rinse with the medium that you kept in order to obtain the maximum mitotic cells, and centrifuge them at 1500 rpm for 7 minutes. Now, we proceed to the last step. we discard the supernatant by pouring. Then we will resuspend the cells into the medium, which is DMEM without fetal bovine serum and supplemented with colchicine. Plan 2 ml perfusion. Keep the cells ready in ice. Now that both cells are ready, we can start the actual fusion process. We have resuspended the lymphocytes in 1 ml of medium and the CHO cells in 2 ml. Remember that for one fusion we need the quantity of lymphocyte present in 1 ml of blood and the equivalent of 1 T75 of confluent CHO cells. We take the mitotic CHO cells and mix them with the lymphocytes in a round bottom tube. The shape of the tube is important for the next steps. Then we centrifuge and discard the totality of supernatant. This step is tricky, so pay attention. After having discarded the supernatant by pouring, we reverse the tube and let it dry on paper for a few minutes. To induce the fusion, we add 150 microliters of polyethylene glycol on the cell pellet in the inverted tube. While injecting, we return the tube to an upright position. Then we add gently 1.5 ml of RPMI without FBS with colchicine and mix by rotating the tube by hand as shown. We can centrifuge at 1200 RPM for 7 minutes. Now we proceed to the last step before the fusion. We discard the supernatant and reverse the tube on a tissue to let it dry. Then we add 0.7 ml of preheated at 37 degrees fusion medium while rotating the tube by hand. The composition of the fusion medium is indicated earlier in the solution list. For the fusion to occur, we incubate for about 80 minutes at 37 degrees in the cell incubator. After the fusion, we proceed to the cell fixation. Before starting, the potassium chloride solution, prepared at 75 millimolar, needs to be heated at 37 degrees. After homogenization, we carefully mix 10 ml of preheated potassium chloride with the fused cells. We homogenize by pipetting carefully. Then we transfer the whole content to a 15 ml tube. We incubate in a water bath set up at 37 degrees for 10 minutes. Next step is the prefixation step. Make sure to prepare the fixative just before using by mixing 3 volumes of ethanol with 1 volume of acetic acid. For prefixation, we homogenize and add 2 drops of the Carnoy 2 fixative, equivalent to 100 microliters. After inverting the tube for homogenization, we centrifuge at 1400 rpm for 8 minutes to obtain the cell pellet. The following step is the actual fixation, still using the Carnoy 2 fixative. After centrifugation, you can see the small cell pellet. We carefully discard the supernatant. Feel free to keep a few liquid to avoid loss of cells. Then we add 10 ml of the Carnoy 2. It is important to proceed as I am showing in the video. We alternate, adding the fixative solution and mix it gently by tapping the tube. Avoid making violent moves since they could lead to Y chromosome spreading and make the analysis impossible. Finally, we centrifuge. We need to repeat the previous step and fix a second time with the Carnoy 2 fixative. The fixed cells can be stored in the Carnoy 2 fixative at this stage up to 1 week at 4 degrees. Chromosome in metaphases will be spread on slides for staining and microscope imaging. The preparation of the slide is a very critical step and can really impact the quality of the acquired images. Microscope slides need to be cleaned one by one, first with tap water, then in a beaker using distilled water. After that, they can be kept in distilled water at 4 degrees up to 1 week. This step is usually performed the following day after overnight fixation incubation. Fixed fused cells are centrifuged and washed with Carnoy 2 fixative, at least one, depending on the cell pellet appearance and the presence of aggregates. Again, be prudent while mixing the cell with the fixative and make moderate tapping on the tube. Another thing is that you need to prepare a fresh solution of Carnoy 2 fixative. Cells are now ready to be spread on slides. After our last washing, we will keep about 2 volumes of liquid above the cell pellet as I am showing you. We will suspend cells and homogenize by pipetting. We take 20 microliters of the cell suspension to spread it on a slide covered with water trickle. We try to distribute the suspension uniformly on the wall length of the slide. Wait a few minutes until the slide has dried. We check with a microscope if the cell concentration is adequate for the analysis. If it is not the case, we can add more drops of fixative or centrifuge to then adjust the volume. Here in Procitox, we make sure to carefully label and mark each slide. The slide can then be stored in boxes at -20 degrees for years or at 4 degrees for a few weeks. Before acquiring and analyzing the data, you need to perform DNA staining. You have two main options. The classic and conventional one that we rarely use in the ProCitox laboratory is the Giemsa staining. This staining is easy and fast. Then the slides can be directly visualized using an optical microscope. In the case of PCC, it only allows the counting of excess PCC fragments. Nevertheless, our favorite one is the centromere and telomere fish staining that facilitates the analysis after acquisition. This staining method will be the topic of another tutorial that I recommend you to watch if you plan to perform the technique. When applying to PCC, it allows for the scoring of decentric and ring chromosome, as well as acentric fragments. Here you can see the same PCC after Giemsa and centromere and telomere fish staining. The only inconvenience of the fish technique is the intensive staining of interstitial telomere sequences in hamster chromosome. In CAA, we are specialized for the method of centromere and telomere fish staining. So we improve the technique for PCC to avoid the problem of the intratelomere staining. Instead of using the hamster CHO cell line that is conventionally used, we establish a new cell line from the Akodo rodent. We select this species because it has a low number of chromosomes, highly dysmorphic as regard to human chromosome, and very few interstitial telomeric sequences. The fusion protocol is similar, and the use of these cells does not require any different steps in the analysis.
Comparing to the DCA and CBMN assay, the PCC assay looks more complicated. Can you tell me what are the advantages of performing this technique?
Yes, that is true. It takes one full day of work. But you don't need to wait for establishing a cell culture and for the lymphocyte entry in mitosis, as it is the case with the two other assays. And it allows the detection of much higher dose due to the absence of mitosis that can be impaired in case of very high DNA damages.
So it means that you receive the blood on one day and you can have the data just the day after?
With the Giemsa staining, I can say yes. If you make a centromere and telomere fish staining, maybe you need one more day to proceed to the staining and to acquire the images. So, less than 48 hours later, you will get the result.
Okay, so the different steps are in order: isolation and fusion with lymphocytes, fixation and spreading on slides. Then we perform the desired staining, we acquire the images, and we analyze. For performing the analysis, do I need to be a specialist?
Giemsa analysis require less experience. I mean, you just need to distinguish between hamster and human chromosome, and then you just count human chromosome fragments. The centromere and telomere fish staining of PCC is quite infrequent. But if you know how to analyze the staining for DCA, for example, you will have no difficulty. I mean, it is quite similar. You just have to be able to distinguish decentric chromosome with two centromere in green or acentric fragment without any centromere. The only difficulty is that the CHO chromosome contain intra and repeated telomere sequences that lead to important telomere staining in red. Thus, many fusion with the intrication of human and hamster chromosome cannot be analyzed. Now, with the modification of the protocol and the use of Akodon cell, we will not have this problem anymore. However, the total number of fusion is lower with Akodon cells.
For me, the analysis doesn't seem so straightforward, right?
You know, we have developed a plugin for ImageJ to guide people for the analysis. Using it can really help. And maybe for the first time, you can participate to an intercomparison training between biodosimetry labs. We organize such exercises quite often to address the issue of the confidence of results. In this way, you will be able to compare your results with others.
Okay, so it seems that the PCC is a quite fair way to determine doses. What is its sensitivity?
Of course, this information can differ between the study, but is usually considered to be about 50 mg, which is quite low. We are exploring the possibility to increase the sensitivity of this assay with the centromere and telomere fish staining.